We use this assay to assess whether plant foods contain phytosteroids, which can affect physiological processes in animals, as well as behavior and reproductive success. This assay provides a simple and reliable method for determining whether plants produce estrogenic compounds with biological activity.
This method was developed initially to screen drugs with xenoestrogen activity for applications in medicine. For example, inhibiting cell proliferation. We are using it to provide insight into primate ecology and evolution. It is important to have practice working with aseptic technique in a clean environment prior to attempting this assay, as it is very sensitive to contamination.
Begin by freeze-drying the fresh plant items with a lyophilizer, then finely grind them using a grinding mill with a 0.85 mm mesh screen. Store the ground samples in bags in the dark until extraction. To extract any potential phytosteroids in the plants, add dried sample to an appropriately sized flask, then add HPLC-grade methanol. Cover the plant methanol solution with aluminum foil, and set it to rotate at 100 rpm for 3 days on an orbital shaker. After 3 days, use filter paper to decant the supernatant into a drip filtration system. Dry the plant extract with a rotary evaporator until it is thickened but pourable in a 300 ml round bottom flask. Pour the sample into a 50 ml flask and rinse the large flask with a small amount of methanol, then continue to dry the sample in the small flask until the methanol is completely evaporated. When finished, weigh the sample residue with an analytical balance and record the mass. Dissolve the plant extract in DMSO at a concentration of 0.1 gram of